Genetics in the Real World, Colorpoint Pattern in Siamese Cats by Lauren Allen. The colorpoint pattern consists of darker colors on the face, ears, feet, and tail, and there is a paler color on the body. The point pattern is most commonly associated with Siamese cats, but it can also appear in any domesticated cat. This pattern is not only limited to cats, but it can also be found in rabbits, horses, rats, and sheep. Many people tend to recognize Siamese cats by the traditional dark brown seal point pattern. However, there are other color point patterns such as the chocolate, blue, lilac, flame, and torty point. The point coloration in Siamese cats is a form of partial albinism where their eyes are also affected. Because of the restriction of pigment in the color point pattern, the eyes of Siamese cats are always a shade of blue. A temperature-sensitive allele is an allele that's expressed only at certain temperatures. In lower temperatures, the allele is expressed, however, in higher temperatures it's not. The color point pattern shown in Siamese cats is created by the Siamese allele, and the expression of this allele is temperature-dependent. This Siamese allele produces the tyrosinase gene, which is necessary for the production of the dark pigment, and the tyrosinase enzyme is inactivated at higher temperatures. The darker pigmented areas of a Siamese cat is restricted to the face, feet, ears, and the tail because the cat's core body temperature is around 100.4 to 102.5 degrees Fahrenheit, and the tyrosinase enzyme is functional only in the cells of relatively cool extremities. Now, as shown on the images to the right, you may wonder why a Siamese kitten is all white in color compared to an adult. Siamese cats are born all white because the tyrosinase enzyme is inactivated due to the mother's womb being uniformly warm. As the kitten ages, the darker pigment develops on the cooler regions first. The torso then becomes cooler with age, and the body eventually darkens. A missense mutation alters the coding sequence so that one amino acid is substituted for another, where the substitution of one amino acid for another is the result from a change in one DNA base pair. The mutation that causes the color point pattern in Siamese cats changes the amino acid from glycine to arginine in the tyrosinase protein, and is therefore a missense mutation. The change in one DNA base pair, guanine to adenine, changes the amino acid from glycine to arginine, and this causes the color point pattern in Siamese cats when both copies of their DNA has the same change. For example, let's look at the DNA sequence in the area around the point mutations. The top shows the DNA sequence of my non-pointed cat Sophie, and the bottom shows the DNA sequence of a pointed Siamese cat. You can see that the single base pair change of guanine to adenine results in the substitution of glycine to arginine, and this represents a missense mutation. A SNP is a DNA sequence variation occurring when a single nucleotide, such as adenine, thymine, cytosine, or guanine, differs between members of a species or paired chromosomes in an individual. SNPs generate biological variation between species, causing differences in the recipes for proteins that are written in the genes, and these differences can influence many traits such as appearance. For example, when we go back to the DNA fragment of my non-pointed cat Sophie and the DNA fragment of a Siamese cat, we can see that they contain a difference in a single nucleotide, and we can say that there are two alleles, G and A. The change of guanine to adenine, which in turn changes the amino acid in the tyrosinase protein, is a SNP that causes the color point pattern in Siamese cats.